Hi friends, how are you? Welcome to my YouTube channel D&D Pathshala. In this video, we will discuss regarding the detailed information of the chromatin, telomere and the remaining part of the chromosome. In the previous video, we have studied regarding the structure and the basic structure and also the general characteristics and the autosomes and allosomes. So in this video, we will start from the chromatin. So let's get started with our video. As we have discussed the differences between the chromatin and the chromatid and the chromosome, here the differences of the chromatin and chromosome are present. So actually a chromatin will be consisting DNA and RNA and also the proteins. So these proteins will be having histone proteins and non-histone proteins. So these histone and non-histone proteins will be seen in the further slides. As we all know, DNA is one of the most important type of chemical component and it will be playing a very important role. Like it will be passing on the hereditary and also controlling the heredity. So it is one of the most conveniently type of measured. So the units are picograms. So we can measure conveniently by these units. And also in addition to this, to describe any kind of genome of an organism by its number of the chromosome, so it is also described as the amount of the DNA that which are present in a haploid cell. So we can estimate it by the number of the chromosomes. And also in the eukaryotic chromosomes and also not only the eukaryotic type of chromosomes. So these eukaryotic type of chromosomes will be present in the organisms. So in the eukaryotic organisms, they will be having 2 to 10 times as many genes. Means their chromosomal rate will be having more. So genes will be 2 to 10 times more than compared with the prokaryotic organisms. And also the lower eukaryotes, means the eukaryotic lower classification, will be having less DNA like nematodes. And also these nematodes, like for example Cenorhabditis elegans. So Cenor type of Cenorhabditis elegans, which is having 20 times more DNA than compared with the Escherichia coli. And the vertebrates are having more amount of DNA. It is about 3 picograms. Means it is 700 times greater than the E. coli. And also among them, the salamander amphiuma. So this salamander amphiuma is having a greater DNA content, that is of 84 picograms. And when compared with a man, so man is having 3 picograms of DNA per each haploid genome. So what are these histones means? Histones are the actual type of basic proteins, as they are enriched with basic proteins, arginine, lysine. So these are the amino acids. And by these it will be forming a highly condensed structure. So these histones are again of 5 types. One is H1, H2A, H2B, H3 and H4. So these are very similar among these different species of eukaryotes, and these should be actually conserved. Means for highly conservation process is required during the evolution. So H1 histone type, it is one of the least conserved among all and it is loosely bound to the DNA. So H1 histone is absent. So it is present in mostly least type of conserved and it is present in the different species. Whereas this H1 histone is absent in the Saccharomyces cerevisiae. So when coming to the non-histone proteins, these are the additional type of component different proteins to the histones that which are present in the chromatin. So these will be involving in a different type of activities like DNA replication, gene expression. So there is a scientist by the name Roger Kornberg. In 1974 year, he has described the basic structural unit of chromatin. So this is called as the nucleosome. So this is very helpful for the formation of the chromosome. So now coming to the differences. Now when coming to the differences, the chromatin will be appearing in the interface of the cell, that too in the cell cycle. And when the chromosomes, they will be appearing in the metaphase. So the chromatin will be actually, so the gray part is the chromosome, the green part is the chromatin. So chromatin is present in the interface of the chromosome. So chromosomes will be appearing during the metaphase and they will exist in the anaphase also. So these chromosomes will be having nucleosomes. Chromatin will be having chromatin fibers. So the chromosomes are condensed 50 times than the normal double helix DNA, and they are having thin, long, uncoiled structures, single type of fibers. So these are some of the characters. When compared with the chromatin, So chromatin is actually condensed 10,000 times than the normal DNA, and it is having thick structure, ribbon-like structure. They will be acting as and existing as a pairs. So when coming to its functions, the chromosome will be allowing the genetic material to be packed into a nucleus. That is while regulating the gene expressions. Coming to the chromatin, it will be ensuring the proper type of arrangement of the genetic type of material. Like in the cell type of level, so the equal separation will be ensured with genetic material that which is actually present between two cells. Now the major functions are the chromosome will allow the DNA replications, and it will also allow in gene expression, recombination. Whereas when coming to the chromatin, it is not having any kind of metabolic activity. So this is the information regarding the chromatin. Now these are some of the nucleobases of the DNA. So these are the differences, that is the DNA is having double helical structure and the RNA is having only one helical structure. So some of the amino acid type of proteins, thymine, uracil, cytosine, adenine, guanine. So these are the nucleobases of RNA and these are the nucleobases of DNA. So the difference between these are the differences between histone and non-histone proteins. So histone proteins are H1, H2A, H2B, H3, H4 and H5, and non-histone proteins are of polycombed DNA polymerase, and histone proteins will be associating with the DNA. So non-histone means it will be remaining after the removal of the histones. So histone protein function means it will be help in packing the DNA into nucleosomes, and non-histone protein functions are playing a role in the functions that which are related to DNA. So these are the differences and the structures. So these are the differentiated structures. So this is the DNA. So it is of 2 nanometers. So after DNA, these are the nucleosomes. So from the nucleosomes, chromatin. So this will be acting as a flow chart. So DNA, so if these are being entangled, nucleosomes. So if those are entangled, chromatin fibers. So if these are chromatin fibers are also stretched, then they will lead to chromatin loops. So if the chromatin loops are also stretched, it will be like a condensed type of chromatin loops, like a type of singular type of ribbon layer. And if these are also type of form, then these are the chromosomes. So from the chromosome, so these are the elevating type of structures, chromosome, loops, and if the loops are combined, chromatin loops, chromatin, nucleosomes, and furtherly DNA. So this is the type of flowchart type of characterization. Now coming here, there are actually two important components that are chromatin, and these are also known as the different type of parts. So two important components are, one is euchromatin and another one is heterochromatin. So euchromatin means, if for example if we dye them or if we stain them with the basic dyes, there will be lightly stained regions in the chromosome. So they will be having a single copy of genetically active DNA. So each will be having a single copy. So their function means they will be playing an important role in regulating the gene expression and also in the cell cycles, that is in the interphase. So if these are decondensed, then they are known as the euchromatin. These will be leading to the gene transcription, DNA replication also. So heterochromatin means they will be highly condensed and they will be having their ordered, means settled type of regions in the nucleosomal part, and they will be having highly density means that is highly densitive type of dense type of repetitive DNA. So at the centromeres, that is at the center, and also at the telomere type of forms. So these heterochromatins were about 10% of the interphase chromatin will be heterochromatin, and these heterochromatins are also again of two types. One is constitutive and another one is facultative. So these regions that which remain condensed throughout the cell cycle, means if they are within, so then they are constitutive. Whereas when the heterochromatin condensation occurs, these are known as the facultative. So this constitutive type of heterochromatin will be having very few genes. So these will be leading to the nearby genes transcriptional inactivation process. So such type of gene slicing means these are replicating and these are inactivating. So such type of gene slicing or silencing procedure or phenomenon is known as the position effect. So these constitutive heterochromatin will also inhibit, means there is also another function, that is it inhibits the genetic recombination that which are present between homologous repetitive sequences. So these homologous repetitive sequences will be doing these for DNA duplications and also deletion. So these are the functions. When compared with the facultative, so these are a chromatin. They will be in an inactivated state, that too in the cell cycles during the certain phases of organism's life. And at certain types, such type of, for example, if the heterochromatin, it is a type of dosage type of compensation, like of the X chromosome, or X chromosome inactivation will be occurred in the mammals. So these are the different parts and the functions of the chromatin. So these are the euchromatin and heterochromatin. So these are the upper part heterochromatin and these are the euchromatin. So these are the differences between euchromatin and heterochromatin. So location will be inner body. These are the euchromatin and these are heterochromatin. So inner body euchromatin. So edge means nucleus. Edge of the nucleus means heterochromatin. So only one type euchromatin, and heterochromatin means facultative and constitutive, as I have said in the previous slide. So these are their genetic activities. Now coming to the genetic activity, it will be the exposed chromosomal crossover and genetically active, and here the heterochromatin will be genetically inactive. So euchromatin will be not sticky in nature, heterochromatin will be sticky in nature. So when coming to the functions, it will be allowing, that is the heterochromatin will be allowing the gene expressions to regulated, and it will be maintaining the structural type of integrity, whereas the euchromatin will allow the genetic changes to occur and it will take place, and then the genes are transcribed. So these are some of their presence and functions of the differential type of characters that which are present between euchromatin and heterochromatin. So coming to here, centromere and telomere. So what are the centromere and telomere means? So centromere means these are the condensed type of regions within the chromosome. So centromere and telomere type of, so these are the centromere. So that is which is present in the center. So centromere means these are the condensed regions that which are present. So it will be actually differentiating the upper part and below part, that is the P arm and Q arm, which I have discussed in the previous video. So these are the condensed regions that which are responsible for the acute type of segregation of this replicated type of chromosomes during which processes, during mitosis process and meiosis process. So it is a darkly stained region and its actual location will be present. So where the attachments means this spindle type of fibers will be there. So these are known as the kinetochore, and these will be having DNA, proteins and such type of DNA sequences. So such type of DNA within these regions DNA sequence are known as cDNA. So cDNA, these will be having several type of domains like CDE1, CDE2. So these can move from one chromosome to another, and their length is about, and they are having a length of about 120 base pairs long. Now coming to the telomere, it is a region of the DNA. So at the end, each linear type of eukaryotic chromosomes, so these are required for the replication and for the stability also. So for the such type of stability of the chromosomes, and if any two chromosomes breaks in their cells, means broken in the cells, their ends will be sticky. So by their sticky nature, one end will get attached to the another. So when coming to the human telomeres, they will be containing a sequence like TTA triple G, that which is actually repeated from 500 to 5000 times. And also there will be type of like certain type of bacteria that which will be possessing their linear type of genetic material, like two types. So one of their type will be like a hairpin type of model. So hairpin telomere is the name that which is given to such type of telomere, and by its name, here the telomeres will be bending across from the one end DNA strand to another end. So complementary type of strands. So another type of telomere is known as inverton telomere. So inverton telomere means this type will be actually allowing to overlap between the complementary type of DNA strands. So these are the basic known information and the information that should be known regarding centromere and telomere, and these are the differences. So these are found in the middle of the organism, centromeres. Telomere is found at the end of the organism. Center means centromere, end means telomere. So monocentric type of organisms are also containing single centromere. Holocentric will be having more than one, whereas a single sister chromatid will be having two telomeres at two ends. So these are actually composed of double-stranded heterochromatin DNA, and these are having single-stranded. So here the functions means these will be holding two sister chromatids, and telomere will be protecting the genes at the end regions from getting degraded. So these are the differences. Now coming to the telomere replication. So telomere replication, it is one of the important aspects that which should be known in DNA replication. So what is the primary difficulty that we will be facing with telomeres means the replication of this lagging strand will be a very difficult stage. Means because why it is caused means during the DNA synthesis it will require actually a RNA template. So this RNA template will be providing 3-hydroxyl group type of ion. So by providing such type to the prime type of DNA replication. So actually and eventually the template will get degraded. So if such type of degradation occurs, then it will be a problem. And also a short single stranded region will be left at the end of chromosome. So this region would be susceptible to the enzymes and it will get degraded by a single stranded DNA. So then the result will be the chromosome length will get short after each type of division. So here this is the process. So the replication is one of the difficulty. So this is the process of the telomere replication, and this is also actually known as the end replication problem, which is not actually observed. So this telomerase was actually discovered by the scientists Elizabeth Blackburn and Carol Grader of University of California in the year 1984. So this is the telomere replication. So these are the steps, that is the telomerase enzyme, from that elongation and from their translocation and then elongation. So this is the telomerase replication, and these are, so here telomerase will get associated and RNA template is used and it will get shift and the process will get repeated. So it is a repetitive process. So coming to the human chromosome, the human chromosome is having about 3 into 109, means it is one of the length. So 3 into 10 power 9 or 3 into 109. So these are some of the base pairs. So 3 into 10 power 9 base pairs of DNA it will be present, and the smallest human chromosome, it is actually several times larger than entire yeast genome. So and its extended length of the DNA, it will make up about, the human genome will be about 1 meter long. Means if the extended length of a DNA is stretched, it will be about 1 meter long. And as we all know, the human chromosomes are of 23 pairs, 22 pairs are of autosomes, and 23rd pair is a sex determining chromosome or gender determining. It is also known as allosome. And if the XX is present, then it is a female, and XY means that is heterozygous, that is male. So when they are actually stained, the mitotic type of chromosomes will have a banded type of structure. So these will be actually identifying the each chromosome's karyotype. So each band will be having a millions of DNA nucleotide pairs also. So which do not actually correspond with any kind of functional structure. So by their presence, so these are the informations of the human chromosome. So these are the different type of pictures, that is the human chromosomes, and these are the banding type of pattern, that is by cytogenetic type of staining. So these are the actual type of 23 pairs. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 pairs. So 9 twos. So these are 18 and these are 18. So we can actually get, and these are the other chromosomes. So these are the metaphase type of, so these human chromosomes are human metaphase chromosomes. So there are actually human chromosomes are of types also. So based upon their positions. So based upon the centromere position, the chromosomes are actually divided into metacentric, submetacentric, cosmetacentric, and acrocentric and telocentric. So depending upon the position of the centromere. So if the centromere is correctly at the center and the 4 chromatids are equal length, then it is metacentric. If the centromere is little away, means if it is deviated from the so from the center to either below or it will going either upper, so it is a submetacentric. So here in the submetacentric chromosomes, one side will be slightly longer than the other side. So in the acrocentric means, so the centromeres will be located very closer to one end of the chromatid, means it will be situated upper side of the chromatid or the below side of the chromatid. So that's why the opposite side chromosomes will be in a very long and also in the small round structure. And these are actually attached by a thin thread. So these can be actually observed on the side of the short chromatid. And that smaller round structure, that which is part of this chromatid, it is actually termed as a satellite. And those thin strands of that satellite region are known as the nucleolar organizer region. So these are the acrocentric chromosomes. And telocentric chromosomes means Here the centromere will be placed at the one end of the chromatid. So means one arm, not both the arms. So actually telocentric chromosomes are not seen in the human cells, but these telocentric chromosomes are very rare. So these are the types. So these are the actual types. Means here you can see in the metacentric, these are perfect. So they are having the centromeres at the center. So here in the submetacentric, they are deviated from the center to the upper side. So that's why here these are looking somewhat long. Whereas when coming to the acrocentric, so these are very long arms and the upper sides are very short. So these are actually known as the satellite type of region, and here thin fibers are not actually seen. So thin fibers are nucleolar regioning type of organizer regions. So these are the telocentric type of chromosomes. So these are the types and parts of the chromosomes and the components and their functions. And thank you so much for listening to my presentation. If you like this video, like, share, subscribe, and don't forget to comment. So if you comment, I will be able to answer all your doubts. So please don't forget to subscribe to my channel and please comment. And if you have any doubts regarding any previous video, please comment me in the comment section below. And thank you.